so the focus of lesson 5 is not only learning what all the parts of the cell do, okay, and on a quiz or a test, I'm not looking for a long, detailed description of what they do, okay? Like, let's say on a quiz, you were telling me, like, one of the things was the Golgi Apparatus, okay? So you'd write Golgi Apparatus, and then you could say processes wastes, or gets rid of wastes, okay, or something like that. As long as you have a, 3-4 word answer for what it does, that's good enough, right? Don't write a whole paragraph about how it, there's a cis face and a trans face and stuff comes in one side and goes out the other and it's packaged in vesicles and they go to the membrane. And that amount of detail is not necessary, okay? If I'm just asking what's the basic function, it gets rid of waste. That'll be good enough, right? If it says, write a detailed description of what the Golgi apparatus does, then, okay, then you can include all that other stuff. But if it's just a basic one, it's like a fill in the blank kind of thing, all I'm looking for is it gets rid of waste. Okay, everybody follow there. Okay, so first key point, we need to understand the significance of compartmentalization of cells. All right, compartmentalization is really important, okay? And we only see it in eukaryotic cells. Eukaryotic cells are cells that have organelles. Okay, so our cells are, we're a eukaryotic organism. All of our cells have organelles. A bacterium, on the other hand, is a prokaryotic cell. It does not have organelles, doesn't have a nucleus. Okay, there are no membrane enclosed organelles. We'll talk a little bit more about the advantages and disadvantages of that at a later date. Okay, but for now, know that we're going to be talking almost entirely about eukaryotic cells. That's the ones that have compartmentalization. Okay, we need to know the role of each organelle, and again, that'll be for the purposes of labeling and telling me the function of things on a quiz or a test. Okay. Thirdly, understand that cells are complex systems of interdependent processes. Interdependent, not independent. Interdependent means that they depend on one another. All right. Any one link in the chain gets broken, and the whole thing will fall apart. Right? They are all interdependent on each other. A chain might be too simplistic. That would imply that it's linear. Right? It would be more like a pretzel, right? Where it can go in any direction and any part of it breaks and there's no continuity between the processes. So they are interdependent. Fourth thing, understand that the cell, or understand how the cell carries out its functions. What point of the cell theory is that? Two. Okay, second time you've seen that point. So, okay, it's definitely important. And then understand how materials are transported across the cell membrane and within the cell. Okay, that will be probably the second most important thing, okay, in today's lesson, is how the cell transports material. All right, so compartmentalization of cells was important because it allowed cells to become larger. Okay, it also allowed them to become specialized, right? So we'll add that as well. We have a whole lesson later on what specialization is and why it's important, okay? But it did allow them to become specialized as well. And that is the kind of main difference between both prokaryotes and eukaryotes. Prokaryotic organisms are all, without exception, single-celled organisms. Eukaryotes can be single cell, but obviously also multicellular. And without specialization, you can't become a multicellular organism. What specialization means is not every cell in my body is identical. Right? My liver cells have way more smooth endoplasmic reticulum in them because their job is to detoxify poisons and break down starches and stuff like that. Whereas my muscle cells have way more cytoskeleton and way more mitochondria because they use a lot of energy and they change shape a lot and all that kind of stuff. Everyone kind of follow me there? Okay, so having different amounts of different organelles is what specialization is, and it allows you to have cells that perform certain jobs. Okay, so prokaryotes, okay, have no compartments and are much smaller than eukaryotes. So to give you an idea and kind of scale here, right, this big purplish thing here is an animal cell. We don't even see the whole thing in this diagram. In fact, we see probably less than a 20th of it. Right? It is very, very large compared to the bacteria. Okay? We can see the entire bacteria in this picture, right? And then we have a virus, which is incredibly small. This here is 1/2 of a micrometer. So for getting some idea, okay, on the size of things, right? A bacteria might be 2 micrometers, okay? Maybe somewhere in that range, 1.5 to 2, okay? Whereas an animal cell is, well, it could be 20 to 40 to 100 and some micrometers across, okay, depending on what kind and how big. Okay, everybody follow me on that? Okay, so eukaryotic cells were able to become larger because they had these compartments, and within each compartment, the stuff going on could go on more efficiently because within that compartment were the perfect environmental conditions for that process. Okay, in a bacteria, everything goes on in the same place. All right, so food gets digested in the same place where DNA is kept, in the same place where sugar is burned, okay, and all that, and waste is packaged and all of that. That all goes on in the same place. All right, so that is the equivalent of essentially keeping your house all at one temperature, okay, and not having a refrigerator and a freezer. Okay, your house right now, the way it works is like a compartmentalized cell. You have the area in which you live, which you keep around 21 degrees. You have your refrigerator, which keeps food from spoiling at around 4 degrees Celsius. And then you have your freezer that's about minus 15 or 16 degrees Celsius. Okay? Each one of those environments does its job very well. If I didn't have compartmentalization, I would have to keep my house at about 8 degrees, which would be uncomfortable for me. My food would spoil faster, and I couldn't keep anything frozen. Okay? Not very efficient. Everyone kind of follow? Okay. Being more efficient allows you to get bigger because you're wasting less resources and energy and things like that. All right. Okay, so DNA is used to make materials. So this can be the first thing we're going to talk about. Okay, DNA is stored in the nucleus. That is the nucleus' primary job, is to store your genetic material. Okay. So within the nucleus is just a snarl of DNA, right? How many people know what chromosomes are? I've heard that term before. Okay, so chromosomes are essentially what is made up of DNA. Okay, they are made up of DNA. But when we see chromosomes, it's usually when the cell is dividing and it is packaged all of its DNA in order to effectively get them to go into the right parts of the cell. When the cell is normally operating, you don't see those shapes of chromosomes. They're completely unraveled, right? So if you can imagine, within the nucleus, it's like somebody took a ball of string and completely unraveled it and threw it in a pile and left it there, okay? It is like the most disorganized thing you could possibly imagine. Within the nucleus, the nucleolus has to find on that mess of DNA all the information it needs, and it's got to find it like that. All right, so it's not the most organized system in the world, but the cells manage to make it work. All right, so the DNA is just kind of everywhere within the nucleus. Okay, and from within the nucleus, okay, protein synthesis is controlled. Protein synthesis is not controlled by the nucleus itself, but by the nucleolus. Okay, so make sure you add that in there. Okay, protein synthesis is controlled by the nucleolus. Now, around the nucleus obviously is a special kind of double-layered membrane, because what's inside the nucleus is really, really important stuff. We don't want it to get damaged. We don't want anything coming in there going, ooh, that looks neat, taking some and walking away, okay, anything like that. So around the nucleus, okay, we have this double-layered membrane that has the nuclear pores on it, okay? That's these things here, right, that you can see under an electron microscope. They look like this middle picture, okay? So we see all of these nuclear pores here. Even zoomed in even more, what we see is essentially the almost fibrous nature of the nuclear membrane. So it's a tough membrane, and it has to be because it's got such an important job, right? So if you look at the nucleus up here, okay, it almost looks like it's got like these bumps almost look like acne, but those are the nuclear pores, okay, each one of those. So the nuclear pores can open and close to allow copies of the DNA to get out, okay, or return to the nucleus. The only thing that is allowed through the nuclear pores is genetic material. nothing else. Okay, and that's how, unfortunately, how viruses take advantage of your cell. We'll talk more about viruses tomorrow, okay, but know that that's part of how they work as well. All right, questions on the nucleus and nucleolus? Now, DNA is used to make materials in this way. Okay, so the nucleolus produces RNA, which is a copy of the cell's DNA that contains instructions on how to synthesize proteins. All right, so essentially what we have inside the nucleus, so here's the nucleus, okay, this circle up here. Within the nucleus, there are special enzymes, and you don't need to know their names. They're called RNA polymerase. It's an enzyme that builds RNA. Okay, what it does is it takes your DNA, that's this blue spiral here, and it, like a zipper, pulls it apart. Okay, because it only needs to copy one side. You guys learned in grade 9 that DNA is complementary, right? T goes with A, and A, whatever, right? T goes with G, sorry, A goes with C. Everyone remember that? Okay, so they're complementary. Okay, now when you pull them apart, you copy one side. Okay, so this RNA polymerase, which is part of the nucleolus, okay, copies one side of the DNA, and it sends that copy out. Because that copy is only a copy of one side, it's called RNA rather than DNA. Okay. It works a little bit different. It's a single strand as opposed to a double strand. So this here, this pink stuff that's being built is the copy of the DNA. That will go out through the nuclear pores and it will meet up with a ribosome. Ribosomes can read the DNA. So they read the DNA and they can use the instructions that are there, sorry, read the RNA, sorry, they can read the instructions that are there and they build the protein from there. All right, so a ribosome is kind of like the foreman of a construction site. It can understand the blueprints, it can get people moving on the construction and create an end product. Okay, so you guys should be able to, at least in about that level of detail, tell me how proteins get built. Okay, so DNA gets copied into RNA. RNA goes out to the ribosomes. Ribosomes make the protein. Okay, that's kind of the basic level at which Science 10 needs to know how protein synthesis works. Okay, in Bio 20, you'll learn a little bit more about the RNA polymerase and all that kind of stuff. But for our purposes, you need to know DNA gets copied to RNA. RNA goes out to the ribosomes. Ribosomes build the protein. Okay, essentially what it says here. Okay, and obviously the proteins get built out in the cytoplasm because A, that's where the ribosomes are, and B, raw materials can't get into the nucleus, right? The nuclear membrane doesn't allow anything in, so all the materials needed to make proteins are outside of the nucleus. That's why we have to send that stuff out. Okay, so like we say, basically be able to explain this process in your own words. Ribonucleic acid, or DNA stands for deoxyribonucleic acid. Yeah. What's that? No, deoxyribo. It's like, yeah, hyphenated. All right, and there's different kinds of RNA. There's tRNA, there's mRNA, so messenger RNA, transfer RNA. There's different kinds of RNA. You guys don't need to know that for there, but if you're looking at the diagram, wondering why sometimes there's a letter in front of it, okay, there's different kinds. Okay. Any more questions on the role of the nucleolus in protein synthesis? All right, endoplasmic reticulum. We said this is probably the most important stuff in the cell outside of the nucleus, okay? And there's two different kinds, right? So we have the smooth ER, okay? Inside the smooth ER, lipids are synthesized, okay? Lipids are fats. Fats are crucial. We use them to make membranes, right? So all membranes, nuclear membranes, cell membranes, whatever, okay? They are all made of lipids, right? Which is why fat is still an important part of our diet. Okay, we need to have some fat, not as much as we as North Americans generally consume, okay, but we need some fat in our diet. Doesn't matter what kind of fat, it can be animal fat, it can be plant fat, it doesn't matter, okay, but we do need fats because we need to make membranes. If we're not getting enough fats, okay, you'll start to notice problems like skin and hair and stuff like that won't be as healthy, okay, because membranes are breaking down, things like that. So Okay, so lipids are synthesized in there for building and repairing membranes. Complex carbohydrates are broken down. Okay, and drugs and poisons are detoxified. Okay, we also see some hormones produced here, although they are produced as well on the rough ER. Okay, so hormones are also produced there. Okay, and those tasks get performed because the ER has quick and easy access to the membrane. So if something needs to be gotten rid of, like the waste products of breaking down a toxic material, it can be directly funneled to the membrane and be expelled, okay, before it contacts any other part of the cell. Okay, so this is kind of the artist's conception of what the ER looks like. Under an electron microscope, this is what we see. All right, so we see that the rough ER, which is the stuff over here on the right, okay, is kind of more organized. Right, there's almost layers of it, and we can see the ribosomes. Those are the black dots all over it. Whereas this jumbled snarl over here, okay, is the smooth ER. Again, we don't see ribosomes on that. And because of that, it's not as linear, so it just kind of snarls all over itself and fills up a large volume of the cell, actually. Okay. So when you're looking at a cell later this week when we do our cell anatomy lab, you probably will see the rough ER, but you won't see the smooth ER. Okay. The reason you'll see the rough ER is because even though you won't see individual ribosomes, the ribosomes are dense enough that they make it dark. All right, and I think I told you before, it'll look like the nucleus has a goatee. Okay, it might be on both sides, but okay, it'll look like it has almost a beard on one side, okay, because that's the rough ER hanging off of it. All right, it's always attached to the nucleus, whereas the smooth ER goes everywhere, but without the ribosomes, you don't see it under a microscope, or at least a light microscope. Okay, questions on the ER, smooth ER? All right, lipids can be quickly moved to the membranes, okay, because again, the ER, both kinds have quick access to the membrane, so they're making membrane material and quickly shuttling it there to repair the membrane if there's any breaks or damage to it, okay, and travel within the ER limits contact with the rest of the cell, which is good because sometimes toxic materials are broken down there. Okay, and then I don't think we wrote this anywhere, but the prime job of the rough ER, okay, is to transport protein. Okay, questions on that one? Okay, so some of the products of the ER are bound for the Golgi apparatus. So sometimes the toxic materials that are in the ER are actually sent to the Golgi apparatus to be processed before they are expelled from the cell. Okay, and we know that the Golgi apparatus is responsible for the excretion and expulsion of waste products. Okay, so those waste products come in the cis face. Okay, that's this side here. All right, so they come in on this side, they get processed. As they're processed through the Golgi apparatus, they get put into these little vesicles. Right, so it almost looks like the Golgi apparatus is blowing bubbles. If you could watch it actually do its thing, it would look like it was always blowing bubbles, and these little bubbles would come off of it, and inside of those little bubbles, which are made of membrane material, okay, are waste products. Okay, those little bubbles or vesicles will go to the cell membrane, they'll merge with the cell membrane and essentially just pop. Okay, the waste products will be then outside of the cell, and the vesicle will just become part of the membrane. Right, but that's the job of the Golgi apparatus. Okay, so you can see under an electron microscope, it looks like this. So on this side, this is the cis face over here. Okay, you can see that there's no vesicles. Everything's just coming in as kind of grainy and black. But coming off of the trans face, we see a lot of bubbles. Okay, a lot of vesicles coming off of the trans face, and those will go and merge with the cell membrane. Okay, questions on what the Golgi does? All right, lysosomes. What are we going to find? What kind of cell are we going to find these in? Plant or animal? Animal, right? Because lysosomes digest food. Plants don't eat food, so they don't need them. All right. Carnivorous plants are an exception. They still don't have lysosomes, but they can produce the digestive enzymes. They're actually secreted outside of the cell. The digestion does not occur inside the cell. Okay? For a carnivorous plant like a Venus flytrap or a sundew or pitcher plant or whatever. Okay. So lysosomes require strong membranes to protect the rest of the cell from the digestive chemicals and enzymes within them. Right? That's essentially, they're essentially digesting cellular material, so you don't want that stuff to get out into the cell. All right? So lysosomes have that strong membrane and they digest food. So if we were just writing down a quick and dirty kind of explanation or definition of what a lysosome is, digest food. Done. Okay, that's all you need to know. All right. Now, one other thing about the lysosomes. Occasionally, something goes wrong with the cell. Most of the time, the cell can detect that. Okay, if something goes wrong, the cell has this ability to detect that something is wrong with it. If it detects that, it'll kill itself. Yeah, it will, because it knows that if it allows the error to continue, that cell could become, yeah. Okay, so if the cell can detect that something has gone wrong and there's a risk that it could start dividing out of control, the cell will kill itself. Okay, it ruptures the lysosomes. Okay, the animal cells anyway. Okay? We don't know necessarily if cancer occurs in plants, okay? But it does obviously occur in animals. So, okay, if a cell detects that is going on, the cell will rupture all the lysosomes so that the digestive enzymes get out and destroy the cell. Okay? That way the cell can't divide and can't continue to become some sort of malignant mass. Okay? Unfortunately, usually the problem is in the DNA. And if the DNA has a mistake in it, the cell may not recognize that it's doing anything wrong. That's when it becomes a malignant tumor. If it recognizes something goes wrong, then we never know and we stay healthy. Okay? If something goes wrong with the cell's detection machinery, then obviously it becomes malignant, a mass, has to be removed, etc. Right? So the lysosomes do have that extra little function, sort of terminal function, but still a function. All right. Now, mitochondria. Okay, mitochondria are in both types of cells because the fuel for plants and animals is still sugar. Okay, it's just how they get their sugar that's different. So once the sugar is produced, then it's sent to the mitochondria where cellular respiration occurs, and that is the burning of the sugar in the presence of oxygen, releasing energy. All right, because it releases a lot of energy, it has to have a double membrane. Essentially, it acts as an insulator so that the rest of the cell doesn't overheat. Okay? I mean, we all know if we start exercising, running, whatever, we get hotter, right? That's because we're burning more sugar, okay? And essentially, it's from billions upon billions of these little mitochondria running faster, okay? Our body temperature goes up, and as a result, okay, we have to cool ourselves, so we sweat and things like that, and that all helps us to get our temperature back down, okay? If you're running a fever, okay, that is your body's immune system essentially signaling to every cell, we're sick. And one of the best ways to kill an infection is to run too hot. Because most of the things that would make us sick like their temperature right around 37 degrees Celsius. We start running a fever, those organisms, especially if they're bacteria, tend to not function very well, and it's easier for our immune system to kill them off. The problem is that it's not really good for us to be really hot either, okay? But we can tolerate it for a little bit longer than a bacterium could. It gives our immune system essentially a chance to get ahead. Okay, so when you're running a fever because it's your immune system is telling all your cells, fire up those mitochondria, make them run a little bit faster. Let's get the temperature in here up and see if we can weaken the stuff that's making us sick. Okay? Now, sometimes that gets out of control. Okay? Depending on what type of infection you have, okay, there's sometimes a feedback loop that gets created, and your body just continues to send the signal to up your metabolism. Okay? You start getting over 40 degrees Celsius and you start having some serious health issues. Okay? You may start hallucinating, all kinds of terrible things can happen if your body temperature gets too high. I mean, how many people have had sunstroke or heat stroke before? Okay? It's the same thing. Your body temperature was too high for too long. Okay? It's not good for you. Okay? You have the sun hangover after that. Okay? It's awful. Okay, you're dehydrated, you feel rotten. Okay, but yeah, all of that is the result of too high a temperature. Okay, so that can be done on purpose sometimes. Okay, and other times it can be an environmental thing. All right, so if we're writing down a definition of what this does, it burns sugar for energy. Okay, burns sugar for energy, carries out cellular respiration would be a nicer answer, but okay, burns sugar for energy would be acceptable. Okay, any other questions about the mitochondria? All right, chloroplasts. We're only going to find those in plant cells. All right, their job is carry out photosynthesis. If you are writing down a function on a quiz, it's carry out photosynthesis. All right, that's about the only answer I would accept, as that is their job. Okay, they hold the chlorophyll. The chlorophyll is the pigment that allows the capturing of energy through the excitement of electrons, and then the electrons descend and we capture the energy. Okay, so chloroplasts carry out photosynthesis. They also have a double membrane because they're absorbing a fair amount of solar energy, and that can lead to them getting warmer, and they don't want to overheat the rest of the cell. Okay, so this is the artist's impression, and this is what they look like under an electron microscope. You can see that this single organelle, okay, is 3 to 5 micrometers across. Okay, that organelle is bigger than most bacteria. Right, which is why, when we look at the plant cells in the lab, you will be able to see the chloroplasts. They are a relatively large organelle, and they're kind of grayish green, so they'll be obvious. Okay. All right, questions on the chloroplast? All right, other organelles that we haven't quite talked about in a lot of detail. Okay, the cell membrane. Now, to give you an idea, okay, a lot of people get kind of the wrong idea about what the cell membrane looks like. Okay, the cell membrane is not this like Ziploc bag looking thing. A lot of people get this kind of picture of that it's smooth. Okay, the cell membrane is a matrix of many different things. Yes, it's mostly a layer of fats, but embedded in those fats are all kinds of proteins and sugars and other types of like even steroid kind of things, like cholesterol is actually a steroid. Okay, so all of those things get embedded in the membrane. There are receptor proteins, there are transport proteins that happen, that are a part of the membrane that allow things to get in and out. So it's much more complex than most diagrams of the cell would lead you to believe, including the one we labeled, right? It just looked like a smooth layer, and it's not. Okay, so there's like glycolipids, that's a lipid, that's a fat with sugars on it. Phospholipids, okay, are lipids with a like phosphorus tail. Globular proteins, okay, so a big glob, okay, of protein. Okay, then we have helical proteins because they have a spiral on them. Okay, all that kind of stuff. So it's a very intricate matrix of lots and lots of things. Now, the primary job of the cell membrane, okay, is to carry out these two processes. Okay, it controls endocytosis and exocytosis, and I would expect those words as what you would write on an exam or a quiz. Okay, controls endocytosis and exocytosis. If you write down controls what gets in and out of the cell, you're not going to get a mark for that. Okay, you need to use the technical terms. Also, don't write protects the cell. Doesn't do that either. Okay, it separates the cell from its environment, but it doesn't necessarily always protect it. Okay, now the cell wall. I was telling you about this the other day as well, that the cell wall is not a wall like that wall. Okay, it's a frame, not so much a wall. Okay, so within that wall would be a frame. Okay, in this school they're not wood, they're metal. Okay, but in your house they would be wooden frame. It would be a wood frame, 2x6. Okay, lumber. Right. So essentially, it's a frame that goes outside of the cell membrane. So the purple part here is the cell membrane. The green part are the cellulose fibers that make up the wall, and you can see that there's holes in it. Okay, so it's not a wall that is impenetrable, you know, like Trump wants to build. Okay, it's a frame. Okay, so people can walk right through it, and stuff can go right through the cell wall. So the cell wall does not protect the cell in any way. It is not a barrier to things going inside the cell. It is just a frame that supports the shape of the plant cell. Okay, and that's what we have here. It is the cellulose fiber frame of the cell. It keeps the shape. So frame of the cell keeps the shape. Okay, and then we have the water vacuole. Right, it's essentially a water balloon inside the cell. And that pushes everything in the cell against the cell wall, reinforcing, again, the shape of the cell. Okay? That pressure that is produced by that structure is called turgor pressure. Okay? It's pressure of internal water, turgor pressure. Okay? That's going to support the shape of the cell. And we talked about how if you're not watering a plant, it's going to wilt, okay, like this one is. Okay? It's getting all wilty. The leaves are all kind of soft. Okay? So I should probably water it. It's getting very sick. Right? But I usually don't water them before the biology unit because then I get wilty plants that I can show you. So yes, I torment the plants in this room to teach you something. So that's what we see. Okay, now eventually that gets out of control. If I don't water it for too long, then it goes beyond being wilted. To being dead. I forgot about this one over the summer. It died. But I kept it here, thinking of this lesson. Okay, so this one's kind of irrecoverable, right? It's not like soft and spongy, it's just dry and crunchy. Okay, at that point, no amount of water is going to bring it back. Okay, that one can still be saved, right? It's still green. I can still get it watered, okay, and get it to come back. Oh, well, yeah, but it's that kind of dead grayish green, as opposed to like the we're still alive, but save us green. Yeah. All right, so the water vacuole, like we said, uses turgor pressure, okay, to support the cell, okay, from within. We talked about centrioles the other day also. Okay, centrioles are only in animal cells. Right, and they help with when the cell divides. Right, so when the cell is going to divide, and again, this is only animal cells, the centrioles will migrate to the poles of the cell. And when they get there, they will send out little fibers, okay, similar to the ones the cytoskeleton is made out of, that will attach to each chromosome, okay, along what's called the metaphase plate. You don't need to know that, but okay, they all line up in the middle. Right, so they send these fibers there, and the fibers pull the chromosomes apart. Okay, so it'll pull these apart so that each cell gets a copy, okay, and then the cells will function properly. All right, so what that looks like here, okay, the centrioles duplicate, okay, they mature and they separate, or sorry, the centrioles do, and then they go to the poles, and they can be used to do that. All right, everybody all right with that? Okay, so it's all part of cell division. Okay, basically all you need to write for the centrioles would be helps with cell division in animal cells. Okay. All right, and then we don't have a diagram of this, but we did talk about these. We labeled them on our cell diagram, and that is the microfibers and microfilaments. Okay, we had called those the cytoskeleton. All right, so you might want to write beside those cytoskeleton. Okay, their job is to essentially maintain the shape of the animal cell. Okay. How long does cell division take? It depends on the cell, but it's not normally a long process. And once a cell triggers the division process, it goes through like prophase, anaphase, metaphase, telophase, and then it's done. You'll learn all about those in Bio 20, okay? But they're basically what happens during cell division can be divided up into those stages. So there's the copying of the DNA, the migration of the centrioles, if it's an animal cell, the lining up of the DNA, and then the separation of the DNA, and then the actual separation of the cell. So it's broken up into those steps. It can be anywhere from a couple of minutes to an hour. All right, questions on those? Okay. I'm going to give you guys a little bit of a break because I've been talking nonstop and you've been listening pretty good. Okay, I'll give you a little bit of a break before I go into the types of cell transport here. Okay, so you can take out your phone, talk to your friends, give you about 5 minutes. Okay, so cells have to transport a lot of materials. Now, provided a cell isn't really big, and most cells aren't, they don't have to transport anything very far. And that's good because transporting things over long distances is inefficient and difficult for a cell, right? Especially since the majority of intracellular transport, that's transport within the cell, is accomplished by this process, diffusion. Okay? Diffusion is a naturally occurring process that requires no energy. All things naturally move by diffusion down their gradient or down a concentration gradient. Okay, so you can see in the picture here, they've got like a injector that's injecting dye into water. Okay? You can see that the dye is naturally moving away from this area where it's very concentrated to areas where it is less concentrated. Everything naturally does that. Everything naturally moves down its gradient away from an area of high concentration by this process called diffusion. Okay? It is the same process by which if you fart, everyone will eventually know. Okay? Now, if you're really close to the fart, it's unpleasant. If you're really far away, it might just be a, I don't know, did somebody? I'm not really sure. Because, okay, the fart molecules are diffusing throughout the air in the room, and as they move further away from the source of them, okay, they will become less and less concentrated to the point where they will essentially become undetectable. You want that process to happen quickly. Unfortunately, it doesn't happen quickly. Diffusion is kind of slow. All right, but the advantage of diffusion is that it is free. Okay? The cell expends no energy to use this process. It just naturally happens. And since it takes things from areas of high concentration to areas of low concentration, it naturally moves things to where they need to go, at least in general. All right, everybody kind of follow me there? Okay, so you have the place where the stuff is needed. If it's needed there, there probably isn't very much of it. Then there'd be the place where the stuff is made. There would be a lot of it there. So it would naturally diffuse outwards in all directions, but generally towards the area where it is less concentrated. Now, that's not 100% guaranteed. Okay, this isn't Amazon. Okay, it's not, oh, well, it's going to be there in 2 days. Okay, it's not, targeted for this specific address. It's more like a shotgun. Okay, some of the shot will end up on the target and the rest of it will just kind of disperse everywhere. Everyone kind of follow me there? So diffusion is kind of the shotgun approach to cellular transport. But the good news is that cells are small. There's not really a lot of room for them to, spread out a lot before, okay, the material is where it needs to go. So generally diffusion works pretty good, okay, within the small distances that are common in cells, right? So because molecules have intrinsic kinetic energy, okay, or heat, the result of that motion is diffusion. That's the tendency for molecules of any substance to spread out into the available space. So the simple rule of diffusion is, okay, in the absence of other forces, a substance will diffuse from where it is more concentrated to where it is less concentrated. We call that down its gradient. Okay? Diffusion is passive and requires no energy from the cell. Okay? And generally, diffusion occurs through the cytoplasm. I don't think it says that anywhere in here. And if you recall, we labeled that, I think, just on our animal cell, but plant cells have it as well. Okay? Cytoplasm is that liquid material that fills up the volume of the cell. Without it, diffusion would not occur. Okay? All right, questions on what diffusion is? All right, so cells are transporting materials inside themselves. So intracellular transport, this is the process they use. Okay, all intracellular transport is accomplished through diffusion. All right, the next two are about getting stuff in and out of the cell. Okay, mostly in. So osmosis is one of those processes, but osmosis is specific, okay, in that it is specific to only one material. Only one material moves across the membrane through osmosis, and that material is water. Okay, only water. Okay, so anytime a question is asking you, here's a cell, it's doing this with water. Okay, it's moving across the membrane. What method is this? If it's water, it's osmosis. Okay, only water moves across the membrane through osmosis. This is also a passive process. Okay? And the only reason it occurs is because there's differences in concentration of dissolved salts, okay, or solutes on one side of the membrane from the other. Right? Your cells naturally want to maintain a balance between the concentration of solutes inside and outside of the cell. Right? So most of the time, osmosis moves both directions. Okay? It's balancing things out. All right? So it would move equal amounts of water moving in to equal amounts of water moving out. If there's a major change in salt concentrations, then water will move more in one direction. So, for example, is it good for you if you're stranded on the ocean to drink the ocean water? It is not, because the ocean is the world's toilet. Everything flows to the ocean. Remember Finding Nemo? All pipes lead to the ocean. Don't drink the ocean water. It's gross. Secondly, it's full of salt, which is really bad for you. Okay? Because what happens is, if you drink the salt water, you now have way more salt in your blood. Right? Yeah, and so where does it, if now I've got all this blood that's high in salt and all these cells that aren't. So which way is the water going to move? Into or out of the cells? Out. It's going to move out of the cells and into my blood. Okay, because osmosis is passive. Cells can't control it, it just happens. So if I drink a bunch of salt water or eat a bunch of salty food, okay, I'm going to feel thirsty faster. This is why you'll notice this when you get older, okay? But bar food is always really salty. So you drink more. Okay? It's just good business, okay? But bar food's always really salty, okay? So if you consume a whole bunch of salt, you're going to feel thirsty and you're going to get dehydrated. Okay, if you eat or drink too much salt, especially drinking salt water. Okay, you'll get all this salt in you, water will move out of your cells and into your bloodstream, and then your kidneys will detect that you've got too high a volume of water in your blood. So they'll start pulling the water out of your blood, and you'll pee it out. Okay, so now you've just drank a liter of salt water and probably lost 1.5 liters of your own bodily water. Okay, so the process gets like bad fast if you're drinking ocean water, okay, on the ocean. You're actually, this is going to seem gross, but it's true. You're actually better off to drink your own urine than you are to drink the ocean water. At least until your urine gets really concentrated. Okay, if it's like still fairly clear, then it's actually better to drink that than it is to drink the ocean water. It would have less dehydrating effects on you. Now, I understand just how desperate you would have to be to do that, but if you're stranded in the middle of the ocean, it's that or die, okay? If you drink the ocean water, you're gonna die, okay? It's that simple. You will get dehydrated very fast because of this process, okay? Same thing happens with slugs. What happens if you put salt on a slug? They shrivel up because they can't control osmosis either. And they don't have skin like we do, right? You pour salt on our skin, it's not a big deal. Our skin is much different than the outer layer of a slug. So you put salt on a slug, and suddenly all the moisture that's in them is rushing to the outside to balance this sudden input of salt, and they shrivel up. Okay? They also get really slimy. Okay? Because all the water is coming out of them by osmosis, and they can't control it. Okay? Everybody follow me there? Now the opposite is also true. If I'm just like full of water and it's not salt water, okay, then water is going to start moving out of my blood and into my cells because my cells will be higher in concentration than my blood is. Okay, so once you're dehydrated and you start drinking water again, water goes into your cells by the opposite method. Okay, you start getting water into your bloodstream again and your cells can reabsorb through osmosis. So osmosis goes both ways. Okay, it is the passive movement of water across the membrane to balance salt concentrations. In Bio 20, you'll do a lab that looks like this. Okay, you'll have these horseshoe-shaped, like test tubes almost, and in the middle of them, you'll put essentially like a sausage casing. Okay? Sausage casing is made out of intestine, which is cells. Right? So you essentially put a layer of cells separating one side from the other. Right? On one side, you'll put like either distilled water or water like from the tap, and on the other side, you'll put salty water. Okay? Which way is the salt going to move? Well, it's going to move from an area where there's more water to an area where there's essentially less until the concentrations are balanced. Okay? So you'll actually see during the lab the water level on one side go down and it'll go up on the other side. Right? Like against gravity, it'll go that way. Okay? And you'll get equal concentrations of salt on both sides of the membrane. Okay? Making sense? Okay? So osmosis, okay? Water only. Okay, passive transport, water only. Okay, and it's always going to move across the membrane from the hypotonic to the hypertonic solution. Okay, hypotonic means low concentration. Hyper means high concentration. Right, like if you're hyperactive, you're overactive, too active. Okay, so hypertonic means too much salt. Okay. So if we're looking at that from a cell perspective, okay, if we were in a hypertonic condition, that is, I put a cell in really salty water, the cell will shrivel up just like the slug. Okay? Water will move out of the cell by osmosis until the cell shrivels up. Conversely, if I throw a normal cell into distilled water, it will rupture. Because osmosis is not a process that can be controlled, it's a process that happens. If it's in distilled water, is there any salt in that? No. Is there salt inside the cell? Yes. Since there's no way to balance that, none on the outside, some on the inside, water just goes into the cell until the cell pops. Okay? Until the membrane can't hold it anymore and it just ruptures. Okay? So osmosis is not something that can be controlled, okay? But it is something that happens naturally. All right. Questions on osmosis? Okay. The way I would ask about cell transport processes on an exam would be I would give you a real world situation and ask you which cell transport process is occurring here. Explain how this process works and how it applies to this situation. All right, so there would be the knowledge part of that. Oh, I recognize that that's osmosis. Okay, there would be the recall part of that. That is, okay, osmosis is the movement of water across the membrane to balance concentrations. And then there would be the how does it apply? Now that's the tougher part. How does that apply to this situation? Okay, you got to synthesize something there. Okay, but that's how you'd be asked about these. All right, active transport. Active transport is different from the other two because it is active. The other two are passive. They require no energy. Active transport is the cell using energy to pump materials either in or out, but usually in to the cell. Okay. Now, the way it does that is, I showed you a picture of the cell membrane there a while back, and I said it had lots of proteins and stuff embedded in it. Well, one of those proteins is transport proteins or carrier proteins. That's what these are. Okay, so they're special proteins, okay, that are embedded. They are part of the cell membrane. They can change shape. So if the cell expends energy, they can change the shape of these carrier or transport proteins such that they can either close or open to the outside. Okay, so when they apply that energy, okay, so when there's no energy applied, the carrier proteins are open to the outside, right, and stuff can get in them. When energy is applied, they snap shut, but they're open on the inside now. So whatever was captured in between can now move to the inside. When these things really get moving and changing shape, they actually create a pumping, pressurized action that will pull solutes into the cell. The most common solutes transported by this would be calcium, sodium, and potassium. Okay, these are the things that we typically lose in sweat. Okay, and would need to be replenished. Okay, there are also things that we use to send nerve impulses and contract our muscles. Okay, when you contract your muscles, okay, so there's something called the sodium-potassium pump. Okay, and it allows, and it's active transport that just pushes materials back and forth across the membrane, allowing your muscle fibers to change shape. Okay, contracting your muscles. Similar process happens when anytime a nerve impulse is fired. Okay, there's a sodium-potassium pump that moves those ions back and forth across the cell membrane in order to allow the cell to fire nerve impulses. Okay? So it's an important process, okay? But it does use energy, and it uses these carrier proteins. Make sure you remember, make sure you make a note that this is accomplished by these carrier proteins. So active transport is pumping against the gradient. It's moving water uphill. Well, not water, but moving things against their gradient from low to high concentrations. You can see outside of the cell, there's a lower concentration. Inside the cell, there's a higher concentration. So it's going against the other processes. Right? So to pump a molecule against the gradient, the cell must expend energy, okay, meaning it actively pumps a substance across the membrane. Okay, and it does that using carrier proteins. Okay, so the big three methods of cell transport are active transport, osmosis, and diffusion. Okay? Written response questions on an exam would focus on those three methods of cellular transport. Okay? We're going to look at some other methods of cell transport, but they are minor by comparison. Okay? These are the big three. Okay? Questions on active transport? Okay. Can I move on to the next slide? I still see people writing. I don't want to take it away. All right. The rest of these cell transport processes are not like the other one, like the big three. These all involve adding or subtracting from the membrane. So they're very slow. They don't represent large amounts of transport. Okay, so they all involve a change in shape of the membrane. Okay, essentially enveloping something to take it in, or like coming from the Golgi apparatus, okay, that vesicle merging with the membrane and popping, right? So that's what we're talking about here, is transport from the Golgi apparatus to the membrane in a vesicle. That is exocytosis, right? So exocytosis, the cell secretes large molecules by the fusion of vesicles with the cell membrane. Okay, and this is, I've described this process to you a couple of times now when I talked about what the Golgi apparatus does. Okay, so from the Golgi, you have these vesicles, they go and they make contact with the cell membrane, and they just merge with the cell membrane. Whatever was inside of them is secreted, excreted, whatever it is, okay, into the outside, into what we call the intracellular fluid, okay, which is basically the stuff that's between cells. Okay, questions on exocytosis, right? So that's monitored by the Golgi, vesicles and merge with the membrane. Now, that's the only way stuff gets out, okay, through exocytosis. There's three different ways for things to get in through endocytosis. So there's three different kinds of endocytosis. All right, so in endocytosis, the cell takes in macromolecules, that's big things like chunks of food, big proteins, whatever, okay, by forming vesicles budded from the cell membrane. Okay, there's three types of endocytosis. Phagocytosis, that's eating, okay, so taking in solid objects. Pinocytosis, that's just enveloping large amounts of fluid. And receptor mediated endocytosis, that's how your cell takes in hormones and other chemical messengers that may be sent to it. Okay, so you can see here in the top left, okay, we see pinocytosis going on. So the membrane just kind of folds in. Okay? It folds in and then it kind of pinches shut to make a vesicle. Since there's no solid material here, all it has done is engulf fluid. Okay? If the cell is really dehydrated and osmosis is not moving material fast enough, like liquid material, then this is what the cell will do. Right? Phagocytosis is how your cell engulfs solid material. It's also how the amoeba that we looked at the other day eats. Okay? It just engulfs a food particle, okay, encloses it in a vesicle, which will then merge with a peroxisome and become a lysosome. All right, so this process, phagocytosis, if you've ever cleaned up after your dog, Okay, you put the little bag over your hand, right? You pick up the poop and then you turn the bag inside out. That's kind of like phagocytosis. Okay, it's enveloping something and putting it in a vesicle. Okay. And then we have receptor-mediated endocytosis. Again, the mechanics are the same. The cell just engulfs this stuff, but it's actually engulfing its own receptors. We said when we looked at the diagram of the cell membrane that it's covered in receptor molecules. When those receptors receive a signal, they are engulfed and taken in. Okay, and then whatever that molecule, that hormone is supposed to do, that vesicle moves to that part of the cell and triggers the reaction. Right, so if it's insulin, it actually triggers the membrane to be more willing to take in sugar, okay, and things like that. So different hormones cause different changes within the cell. All right, so phagocytosis, the cell engulfs a particle by wrapping what's called a pseudopod, that means false foot, around the object, essentially just engulfs it, okay, packaging it within a membrane enclosed sac called a vacuole. Okay, this is also how your white blood cells kill bacteria. Okay, they eat them, and they just go around and they engulf them and eat them. Okay, so that's phagocytosis. Okay, whatever materials are in there get digested, and the waste products just go back out to the membrane through exocytosis. Pinocytosis is cell drinking, so it's gulping the fluid in. Okay, this is what it looks like under an electron microscope. So this is the cell. Okay, this would be one side of the cell. This is the other side of the cell, and you can see that. You can see these vesicles, there's a whole bunch of them. So they would have likely artificially made this cell very, very dehydrated so that it would gulp in a whole bunch of stuff when they took its picture. And so you can see those quite clearly there. That would be the nucleus. Okay? And it's just taking in a whole bunch of fluid. All right, and then receptor-mediated endocytosis, like we said, the cell membrane is covered with these receptors, okay? And you can actually see them in these electron micrographs. So this right here is a receptor, okay, as is this right here. This right here is a receptor, okay. They're receiving the kind of cloudy looking stuff. This stuff here is the hormones, okay. When they attach to those receptors, then here's the cell membrane. You can see that it's engulfed all of those receptors, okay. You can see them all in here. They each have something sitting on top of them, okay. So these are the receptors down here, okay. The circles are whatever the signal is. They engulf it and they take it in. Okay, that process is fairly rapid, right? Because any chemical signal going to the cell is usually something important, especially if it's something like adrenaline, okay, and it's triggering your fight or flight response. You want that reaction to happen right away. Okay, so again, these are a much smaller part of cell transport. Okay, they're endocytosis and exocytosis, and they involve a change in shape of the membrane. The others do not involve a change in shape of the membrane and actually constitute more of the transport. Okay, so the big three are more important. All right, questions on receptor mediated? All right. Okay, I want you to answer those questions. Hopefully we'll get to them, but if we don't, we'll talk about them tomorrow.